Hello everyone, this is Dr. Jawad. In the short video, I'm going to go over briefly the steps of cellular respiration. Now, just to forewarn you, there's a lot of steps that I'm going to be missing, I'm going to be leaving out, because I want to keep this video very general, okay? So, we're going to start off with cellular respiration. What is cellular respiration? It's basically the splitting of glucose to make energy. Now, it's going to be, the first step is going to be glycolysis. Now, in the presence, is it going to be with oxygen or without oxygen? So, in the presence of without oxygen, anaerobic, there's a process going to go to fermentation, which is going to be creating lactic acid in human beings. Or it's going to go ethyl alcohol and carbon dioxide in plants and yeast. Now, in the presence of oxygen, it's going to go make acetyl-CoA, which is going to go into the Krebs cycle, which the final result is going to go into electron transport chain, and that's essentially where we make all of our ATPs. Now, there's going to be 4 steps. Now, I just like to have a little brief explanation of each. Step 1 is glycolysis. Glycolysis is the splitting of sugar. You're taking the glucose molecule and you're going to split it into pyruvate. Next, step 2, it's called pyruvate dehydrogenase. You're going to take that pyruvate molecule and you're going to further break it down to form acetyl-CoA. Acetyl-CoA is very, very important because that is a segue into the Krebs cycle. And the end result is going to be ATP, NADH plus, as well as FADH2, and it's going to get oxidized to form really the energy needed to bring us into the electron transport chain, which is the final step. Because the electron transport chain, it's very, very complex, and the end result is where we get 38 ATPs. Okay, so if you follow the link below, we're going to go into glycolysis, step 1. Hello everyone, this is Dr. Jawad. In this short video, I'm going to go over the steps of glycolysis, step 1 for cellular respiration. Now, key thing to note, I know there's a lot more steps that I'm missing out, okay? But I'm just going over the basic steps of glycolysis in this video. Now, glycolysis, a couple things you want to understand. One, you're looking at carbon. What is carbon? Carbon is C6H12O6. And in a nutshell, you're taking the 6-carbon glucose, you're splitting it up to make it into a 2,3-carbon pyruvic acid or pyruvate. It's either or, they still mean the same. One thing you want to locate it, where is it located at? It's located in the cytoplasm. It's not within the mitochondria. So just that's a key thing to note. So cellular respiration, glycolysis, step 1, the splitting of the sugars. Now, I like to divide this in 2 phases, kind of like banking. Because phase 1, you have the investment phase, and phase 2, you have the payoff phase. So the investment phase, what does that mean? Off the cuff, that means that you're going to use energy in the form of ATP to split it. So you got the 6-carbon glucose, you're going to use 2 ATP off the cuff to split it, and it's going to become a 2,3-carbon compound with a phosphate group because of the ATP. And this compound is called phosphoglyceraldehyde, or PGAL for short. Now, one thing to note is, again, this has a lot of steps that I'm missing out, I know that. However, this has a rate-limiting step, which is glucose 6-phosphate. Anytime we talk about rate-limiting steps, that's like a safety mechanism so you don't overload the system in this part with the carbohydrate. So you got 2 3-carbon compounds that have a phosphate group, PGAL, and then now you're going to the payoff phase. What's the payoff phase? The payoff phase, you're going to gain something. Okay, so what are we going to gain? Because we want to further split. You're going to gain 2 ATPs. You're going to gain NADH plus. Again, it's going to be reduced to NADH plus. Remember, reduction is you gain a hydrogen. Now, since it's done twice, your net gain, the payoff is you're going to have 2 molecules of pyruvate for every 1 molecule of glucose. So 1 molecule of glucose is going to split into 2 molecules of pyruvate or pyruvic acid. Now over here, what do you produce? Okay, you produce 2 ATPs. You produce 1 ADH for each molecule. Remember, there's 2 of them. So what the net gain is, what you're gaining is, really you're gaining 4 ATPs But remember, you're going to use ATP, 2 ATPs in the investment phase, so your net gain is 2 ATPs. There's 2 molecules here, so you're going to have a net gain of 2 NADH pluses, and also you're going to gain 2 pyruvate compounds. So this is step 1 of cellular respiration glycolysis. If you just hold on, we're going to take you to pyruvate dehydrogenase. Thank you. Alright, this is Dr. Jawad. What I'm going over step 2 of cellular respiration, which is called pyruvate dehydrogenase. Now let's back it up a little bit. So step 1 was glycolysis. You're taking the splitting of sugars, you're taking the split of glucose, a 6-carbon glucose, and the end result is 2,3-carbon pyruvate. So remember, with glycolysis, important note factors are the rate limiting step is glucose 6-phosphate. Where does it occur? It occurs in cytoplasm. And the net gain is 2 ATPs and 2 NADHs. So now, pyruvate dehydrogenase, now what I'm doing, I'm only going to show one of them because there are 2. So you have a step that again, pyruvate dehydrogenase. What it's going to do is going to cleave off one of the carbons off the pyruvate. And also you're going to have a reduction. Remember, reduction is a gain of electrons. So you have an NAD, and it's going to result in NADH plus. So again, one of the carbons are going to be cleaved off, and you're going to have a net gain of 1 NADH plus. So really, pyruvate dehydrogenase breaks off a carbon off the pyruvate, which gives you 2-carbon acetyl-CoA, which this will take us into the electron transport chain. So the net gain of pyruvate dehydrogenase is you're going to gain 2 NADHs. Remember, because here I only showed you 1, but there's 2 3-carbon pyruvates that's going to produce 2 2-carbon acetyl-CoAs that will bring us into the electron transport chain. Okay, so follow the next video. Step 3 is the Krebs cycle. Thank you. Hello everyone, this is Dr. Jawad. I'm going over cellular respiration. I'm going over step 3, okay, the Krebs cycle. So let's just review some of the steps. So step 1 is glycolysis, and the end result is you're gaining 2 ATPs and 2 NADH pluses. Step 2 is pyruvate dehydrogenase, and what you're doing, you're turning the pyruvate into acetyl-CoA, and your net gain is 2 NADHs. So now you're taking the acetyl-CoA, which is very, very important, and that is going to, step 1 is going to merge with oxaloacetic acid. So you're taking the glucose, you're splitting off the 2 3-carbon pyruvic acids, pyruvate, and that's going to turn into acetyl-CoA, a 2-carbon. And this is an important thing. This is going to merge with oxaloacetic acid, a 4-carbon, to form citric acid. Citric acid, just like the orange juice that you eat. Now, it's going to go through a series of steps, and they're all catalyzed by enzymes. Now, key note, I'm just making this very, very basic. I know I missed out a lot of steps, okay? But I just want to keep this basic. So the Krebs cycle spins twice. Why does it spin twice? It spins twice because you have a 2-carbon acetyl-CoA. And it's catalyzed by enzymes, so you're going to go through a lot of reduction and oxidation cycles. So first and foremost, you're going to get NAD. It's going to get reduced to NADH plus. As you're going to have another NAD, it's going to be reduced to NADH plus. You're going to clean off 1 carbon, okay, CO2. You're going to generate ATP from ADP. Also, you're going to cleave off another carbon here. You're going to reduce FAD to FADH2. Another NAD plus, you're going to reduce it to another NADH plus. Now, the reason why it's going to spin twice is because with every rotation, you're cleaving off 1 carbon. Okay? Now, you're going to ask yourself, well, you have a 6-carbon glucose that's going to turn into 2 3-carbon pyruvates. Yes. So, how is that going? Here's the first carbon that's going to be cleaved off. And pyruvate dehydrogenase, because that's going to bring into a 2-carbon acetyl-CoA. So again, this is why it spins twice. And the end result for the Krebs cycle is you're going to get 3 NADHs, 1, 2, 3. You're going to get 1 FADH2, 1, and then you're going to get 1 ATP. But wait a minute, don't forget, it spins twice. Why does it spin twice? Because you have to cleave off the carbons. So then we have the end result as 6 NADH pluses, 2 FADH2, and 2 ATPs. Why is this important? This is important it's important because this is going to bring you into the electron transport chain, which the electron transport chain is where we make 38 ATPs. Now, just a few things to note. Again, the Krebs cycle, it's catalyzed by enzymes. Important, acetyl-CoA merges with oxaloacetic acid. It spins twice, and where is it located at? The mitochondrial matrix. Now we're inside the cell. Now, an important thing to note, acetyl-CoA is like the first step inside the door. Because when we're talking about glycolysis, we're talking about carbons, we're talking about carbohydrates turned into energy. However, acetyl-CoA is also the entranceway for fatty acids and amino acids. Fatty acids through beta oxidation and amino acids through gluconeogenesis. So if you note that whether you're taking it from carbohydrates or fats or amino acids, the main step is right here, acetyl-CoA. Okay, so that's the Krebs cycle. Thanks for watching. I'll provide a link down below for the 2 steps before this. Thank you. Okay, so step 4. Step 4 is the electron transport chain. Now I'm going to keep it simple, so I'm not going to go over all these steps. But let's rewind. So we have step 1, glycolysis. What is the end result? You have 2 ATPs and you have 2 NADHs. Then you have step 2, which is pyruvate dehydrogenase. What's the end result? You have a formation of 2 NADH pluses. And then step 3, you have the Krebs cycle. So what's the end result? You have 6 NADHs, you have 2 FADH2s, and we have 2 ATPs. Now, one thing to keep in mind, that the NADH and the FADH2s, they're very important because why? Because they're the input into the electron transport chain. And they're oxidized. Okay, so the electron transport chain, it's still in the inner mitochondrial matrix. Again, it's a series of steps, but I'm just going to keep this very, very simple because I'm focusing more on what is the net result. So the end game with the electron transport chain is you're taking the NADH pluses and the FADH2s, and the end result is for every 1 NADH, you're going to produce 3 ATPs, and for every 1 FADH2s, you're going to create 2 ATPs. So how many is that? So you have 10 ADHs and you have 2 FADH2s. Times 3 is going to bring it to have 30 ATPs. The FADH2s times 2, you're going to have a net of 4 HTPs. Add those up, you're going to create 34 ATPs. However, remember, in glycolysis, you're still going to add up those 4 ATPs to have the to have the last final product of 38 HTPs from the electron transport chain. So I hope you enjoyed the video. I hope this helps. See you next time. Thank you.